Hello everyone! I'm so glad you could join me today. I'm very excited about today's topic. I am Lorma Ibabor, the assigned reporter of this topic. Today, we're exploring proteins and alpha amino acids. Proteins are like superheroes in our bodies, and alpha amino acids are the building blocks that make them up. By understanding the structure of alpha amino acids, we can unravel the mysteries of how proteins work. Let's dive in and become proteins detectives together. So I am going to discuss what is proteins all about. As you can see in your screen, that proteins are one of the most abundant organic molecules in living system and have the most diverse range of functions of all micromolecules. So proteins also play roles in communication between cells, transporting molecules within the body, and even in defending against pathogens. So in a nutshell, proteins are essential for life because they do so many different jobs to keep organism functioning properly. Next is proteins may be structural, regulatory, contractile, or protective. They may serve and transport storage or membranes, or they may be toxins or enzymes. Next is each cell in living system may contain thousands of different amino acids. Because amino acids can be arranged in many different combinations, it's possible for your body to make thousands of different kinds of proteins from just the same 21 amino acids proteins. Each would each with them have a unique functions. Next, their structure like their functions vary greatly. It means that things can look and work in all sorts of different ways. Next, they are all, however, polymers of alpha amino acids arranged in a linear sequence and connected together by covalent bonds. So proteins are made up of smaller units called amino acids, arranged in a specific linear sequence determined by genetic information. These amino acids are connected together by covalent bonds called peptide bonds, forming long chains known as polypeptides. This structure is fundamental to the diverse function proteins perform in a living organism or our body. Next, I am going to discuss the alpha amino acids. As you can see in the screen, it says there that the major building block of proteins are called alpha amino acids. As their name implies, they contain a carboxylic acid functional group and an amine functional group. The alpha designation is used to indicate that these two functional groups are separated from one another by one carbon group. In addition to the amine and the carboxylic acid, the alpha carbon is also attached to a hydrogen and one additional group that can vary in size and length. In the diagram below, this group is designated as an R group within living organism. There are 20 amino acids used as protein building blocks. They differ from one another only at the R group position. So alpha amino acids are the basic building blocks of proteins. Each alpha amino acid has a specific structure. It contains a carboxylic acid group, which is COOH, and an amine group, which is NH2, both attached to the same carbon atom, known as the alpha carbon. This carbon also has a hydrogen atom, an additional group called the R group attached to it. In living organism, there are 20 different types of alpha amino acids used to build proteins. These amino acids differ from each another only in the composition of their R groups. The R groups can vary in size and structure, giving each amino acid its unique characteristics and properties. Here is the general structure of an alpha amino acids.
Good day, ma'am. I am Jolinda D. Albert, your student in AG 206. So these my reports. We have amino acids structure and properties. So there are major categories and subcategories of amino acids. So the first, non-polar hydrophobic amino acids. So una, aliphatic amino acids. Next, aromatic amino acids and sulfur containing amino acids. Second, polar hydrophilic amino acids. So the first, polar in charge hydrophilic amino acids. So polar, the acidic and basic functional groups. So polar in charge, the side change contain here U-tom is O is or is. We have that capable of forming permanent. Next, hydroxyl and sulfoxyl containing amino acids. So these include the hydroxyls and sulfoxyls. We have our two categories. We have serine and threonine and cysteine. Next, sulfur containing amino acids. Next, amine containing amino acids. So amine containing amino acids. So we have glutamine and asparagine. And next, acidic hydrophilic amino acids. Acidic, we have two amino acids. We have glutamic acids and aspartic acids. And next, basic hydrophilic amino acids. So the basic amino acids, we have lysine, arginine, and histidine. We have our contain amine functional group that can be proteinated to carry a full charge. Next, non-polar hydrophilic amino acids. So the non-polar amino acids can largely be subdivided into three more specific classes: that aliphatic amino acids, aromatic amino acids, and sulfur-containing amino acids. The aliphatic amino acids, we have glycines, alanine, valine, leucine, and isoleucines, and proline. We have typically contain bronze hydrocarbon chains. We have complicated structures of leucines and valines. So proline is also classified as an aliphatic amino acids. Contains special properties as the hydrocarbon terminal amine, creating a unique 5-member ring structure. As we'll see in the next sections covering primary structure, we have significantly after 3-dimensionals incorporated protein where folds or trans occur of rigidity of commonly found in regions. So next, the aromatic amino acids. We have phenylalanine, tyrosines, and tryptophan. As their name implies, contain an aromatic functional groups within their structure, making them largely nonpolar and hydrophilic to the high carbon, hydrogen, and content. However, it should be noted that hydrophobicity and hydrophilic hydrophilicity represents a sliding scale. We have each a different of amino acids. We have chemical properties depending on their structure. Have different physicals and sliding scale on each of a hydrophilicity and should denote hydrophobicity. And for example, that hydroxyl I group presents in tyrosine increase its reactivity and solubility compared to that of phenylalanine. We have solubility and tyrosines increase its reactivity. Next, methionine, one of the sulfur-containing amino acids, is usually classified under the nonpolar. So we have hydrophobic amino acids. As the terminal methyl group creates a three functional groups, which generally cannot form permanent dipole with the molecules and retains low solubility. Have containing amino acids, we have terminal methyl group creates three functional groups. The non-polar, so hydrophilic amino acids. In last, this my topic, we have hydrophobic amino acids. We have a structure of the 20 alpha amino acids used in protein synthesis. Our group are indicated by circled, colored, and portions of each molecule. Colors indicate specific amino acids classes. So hydrophobic, green and yellow. Hydrophilic, polar in charge, orange. Hydrophilic acidic is blue. Hydrophilic basic is rose. We have the hydrophilic. We have glycine and alanine, valine, leucine. Ang glycine is gly with G. Alanine, ala with A. Valine, val with V. Leucine with Leu L. We have H and CH3. So, thank you.
Good day, Ma'am and John. My name is Jelmark Vigliarosa, and I'm going to discuss about the amino acid structures and properties of nonpolar hydrophobic and polar hydrophilic of amino acids. So, nonpolar hydrophobic is the nutritional amino acids are a subset of amino acids characterized by side chains that are predominantly composed of hydrocarbon structures like isoleucine, phenylalanine, tyrosine, tryptophan. So isoleucine has a branch chain structure with central carbon atoms bonded to a methyl group and an ethyl group. These branch structures distinguish it from other amino acids and contribute to its unique properties. Phenylalanine has a benzyl side chain that consists of phenyl group attached to the central carbon atoms. This aromatic structure gives phenylalanine its unique properties and enable it to participate in various biochemical reactions. Tyrosine contains a hydroxyl group OH attached to the benzyl side chain of phenylalanine. This hydroxyl group gives tyrosine its unique properties and distinguish it from other amino acids. Tryptophan has an endual side chain which consists of benzene ring fused to a 5-member heterocyclic ring containing nitrogen atoms. Proline is unique among amino acids due to its cyclic structures, where the side chain form a ring attached to the nitrogen atoms of the amino group. These cyclic structures give proline its unique properties and affect its role in protein folding the structures. Methionine contains sulfur atoms in the side chain which consists of methyl group linked to the sulfur atom. These sulfur-containing structures give methionine its unique properties and allow it to participate in various biochemical reactions. The aromatic amino acids phenylalanine, tyrosine, and tryptophan, as their name implies, contain aromatic functional group within their structure, making them largely nonpolar and hydrophobic due to the high carbon hydrogen content. However, it should be noted that hydrophobicity and hydrophilicity represent a sliding scale, and each of the different amino acids can have different physical and chemical properties depending on their structure. Polar hydrophilic amino acids. The polar hydrophilic amino acids can be subdivided into three major classes: the polar uncharged, the acidic, and the basic functional groups. Within the polar uncharged class, the side chains contain heteroatoms that are capable of forming permanent dipoles within the R group. These include the hydroxyl and the sulfoxyl containing amino acids serine, threonine, and the cysteine, and the amide containing amino acids glutamine and asparagine. Cysteine contains the thiol group SH in its side chain, which is responsible for its unique properties, including the ability to form disulfide bonds with other cysteine residues. Serine and threonine is the same, contains a hydroxyl group OH in its side chain, which makes it polar and capable of forming hydrogen bonds within other molecules.
Hi, good morning, everyone. I am Idalina Yulabit, so this is my report about the amino acid structure and properties. So we have polar hydrophilic amino acid. Here is the figure 2.2. Polar amino acids are essential for the proper folding, stability, and function of proteins. Their hydrophilic properties allow them to interact with water and other polar molecules, contributing to the overall structure and function of proteins. Acidic hydrophilic amino acid. Acidic amino acids are important for the proper functioning of proteins and biological processes. Their hydrophilic properties and ability to donate or accept protons allow them to participate in various biochemical reactions and contribute to the overall structure and function of proteins. Two amino acids is the glutamic acid and or glutamate and aspartic acid or aspartate constitute the acidic amino acids. Glutamic acid, also known as glutamate, and aspartic acid, also known as aspartate, are two amino acids that belong to the group of acidic amino acids. These amino acids are called acidic because they have a carboxyl group in their side chain, which can donate a hydrogen ion and aqueous solutions, making them acidic. The carboxyl group is responsible for the acidic properties of these amino acids. Glutamic acid or glutamate has a longer side chain compared to aspartic acid, aspartate or aspartate. It contains an additional methylene group in its side chain. This longer side chain allows glutamic acid to form stronger interactions and participate in more diverse biochemical reactions. This amino acids contain side chains with carboxylic acid, functional groups capable of fully ionizing in solution. Here is the figure token 2 or acidic hydrophilic amino acid. Basic amino acids are a group of amino acids that have hydrophilic or water-loving properties and contain amine functional groups. These amine groups can be protonated, meaning they can accept a hydrogen ion in aqueous solutions, giving them a positive charge giving them a positive charge. There are three basic amino acids: lysine, arginine, and histidine. Each of these amino acids has a unique structure and properties that contribute to their function in proteins. First, we have lysine, has a side chain that contains an amino acid, amino group. This amino group can accept a proton, resulting in positively charged lysine molecule. Lysine is involved in various biological processes, including protein synthesis enzyme activity, and the formation of collagen. Second is arginine, also has a side chain that contains an amino acid. Similar to lysine, the amino group in arginine can accept a proton, leading to a positively charged arginine molecule. Arginine plays important roles in protein synthesis, hormone regulation, and the production of nitric oxide, which is involved in blood vessel dilation. Histidine has a unique structure that includes an imidazole group and its side chain. This imidazole group can act as a weak acid or base, allowing histidine to act as a pH buffer and play a role in enzyme catalysis. The imidazole group can accept or donate a proton, making histidine a versatile amino acid in biochemical reactions. That's all. Thank you.
Good morning, everyone. I am the Group 1 reporter. I am Richelle Rindal. Amino acid structure and properties. Protein structures built from the basic amino acids can be hundreds of amino acids long. Thus, for simplicity's sake, the 20 amino acid used for protein synthesis have both 3-letter and 1-letter code abbreviations. Table 2.1. These abbreviations are commonly used to delineate protein sequences for bioinformatic and research purposes. Table 2.1 Amino acid abbreviation. Amino acid of alanine, 3-letter code is ALA and 1-letter code is A. Amino acid of arginine is 3-letter code ARG and 1-letter code is R. Amino acid of aspartic acid is 3-letter code ASP and 1-letter code is D. Amino acid of asparagine, the 3-letter code is ASN and the 1-letter code is N. Amino acid of cysteine is the 3-letter code is ZYS and 1-letter code is Z. Amino acid of glutamic acid is the 3-letter code of GLU and the 1-letter code is E. Amino acid of glutamine, the 3-letter code is GLN and the 1-letter code is Q. Amino acid of glycine, the 3-letter code is GLY and the 1-letter code is G. Amino acid of histidine is the 3-letter code is HIS and the 1-letter code is H. Amino acid of isoleucine is the three-letter code is ILE and the one-letter code is I. Amino acid leucine is the three-letter code is LEU and the one-letter code is L. Amino acid of lysine is the three-letter code LYS and the one-letter code is K. Amino acid of methionine is the three-letter code MET and the one-letter code is M. Amino acid of phenylalanine is the three-letter code PHE and the one-letter code is F. Amino acid of proline is the three-letter code PRO and the one-letter code is P. Amino acid of serine is the three-letter code is SER and the one-letter code is S. Amino acid of threonine is the three-letter code is THR and the one-letter code is T. Amino acid of tryptophan is the three-letter code is TRP and the one-letter code is W. Amino acid of tyrosine is the three-letter code is TYR and the one-letter code is Y. Amino acid of valine is the three-letter code of VAL and the one-letter code is V. A chiral molecule is one that is not superimazable with its mirror image, like left and right hands that have a thumb fingers in the same order, but a mirror images are not the same chiral molecules that have same things attached in the same order, but a mirror images are not the same. The mirror image versions of chiral molecules have physical properties that are nearly identical to another, making it very difficult to tell them apart from one another or to separate. Because of this nature, they are given a special stereoisomer name called enantiomers, and in fact, the compounds themselves are given the same name. These molecules do differ in the way that they rotate plane polarized light and the way that they react with and interact with biological molecules. Molecules that rotate the light in the right-handed direction are called dextrorotary and are given a D-letter designation. Molecules that rotate light in left-handed direction are called levorotary and give a L-letter designation to distinguish one enantiomer from the other. D and L forms of alanine are shown in figure 2.4b. That's my all report. Thank you.
Represent a quarter presentation entitled Amino acid structure and properties. Alpha amino acids are chiral molecules. Let us know first what is alanine. Alanine is an amino acid that is used in biosynthesis of proteins. It contains an amine group and a carboxylic acid group, both attached to the central carbon atom, which also carries a methyl group side chain. L-alanine are amino acids that are necessary for the biosynthesis of proteins. They are considered non-essential in human diets and those of most other animals because they are synthesized in the body. The D-alanine is an unusual endogenous amino acid present in invertebrates and vertebrates. Compared to its L-isomer, the characterization of a D-alanine is challenging because of the need for chiral resolution and the low amounts of the D enantiomer present. Both L and D split their iron interactive image. Figure 2.4 amino acid chirality in first demonstration is the chirality of the core alpha amino acid structure when the non-specific R group is used. The second is the D and L alanine enantiomer pair. Upper diagram represents the ball and stick model, and the lower diagram represents the line structure. Though most amino acids can exist in both left and right-handed forms, life on Earth is made of left-handed amino acids almost exclusively. Life on Earth is made of left-handed amino acids because there's nothing to prevent life from developing from right-handed amino acids. It just happened that on our planet, the left-handed amino acids were what was available. Meteorites could have delivered left-handed amino acids to Earth, says Levine. Then in turn use the left-handed chirality in developing life. Proteogenic amino acids incorporated into proteins by ribosomes are always in L conformation. Some bacteria can incorporate the amino acids in non-ribosomally encoded peptides, but the use of the amino acids in nature is rare. Ribosomes are macromolecular machines found in all cells that perform biological protein synthesis. Link amino acids together in the order specified by the codons of messenger RNA molecules to form polypeptide chains. However, Doctors John Cronin and Sandra Pizzarillo have shown that of the amino acids that fall to Earth from space in meteorites, more are in the L conformation than the D conformation. Thus, the fact that we are made predominantly of amino acids may be because of amino acids from space. L conformation shows the presence of negative NH2 group on the left-hand side. D conformation means that the amino negative NH2 group is present towards the right-hand side. Why do amino acids in space favor the L conformation? No one really knows, but it is known that radiation can also exist in left and right-handed forms. So there is a theory called the Bonner hypothesis that proposes that predominant forms of radiation in space from a rotating neutron star, for example, could lead to the selective formation of homochiral molecules such as L amino acids and D sugars. L amino acids are the isomer found in proteins within the body. Amino acids are essential to life, have a role in metabolism, and are important in nutrition. Their form short polymer chains called peptides, as well as longer chains that are called polypeptides or proteins. These sugars is overall the most abundant monosaccharide, a subcategory of carbohydrates. This is still speculative, but recent findings from meteorites make this hypothesis more plausible. Meteorites is a rock that originated in outer space and has fallen to the surface of a planet or moon. When the original object enters the atmosphere, various factors such as friction, pressure, and chemical interactions with the atmosphere possible. An argument or statement seeming reasonable or probable. In chemistry, a sweeter ion in a molecule with two or more functional groups of which at least one has a positive and one has a negative electrical charge, and the net charge of the entire molecule is zero at a specific pH. Because they contain at least one positive and one negative charge, bitter ions are also sometimes called inner salts. The charge on the different functional groups balance each other out, and the molecule as a whole can be electrically neutral at a specific pH. The pH where this happens is known as the isoelectric point. Inner salt, a salt formed by reaction within the molecule of a compound having both acid and basic properties. Isoelectric point is the pH at which a molecule carries no net electrical charge or is electrically neutral in the statistical mean. Unlike simple amphoteric compounds that may only form either a cationic or anionic species, a sweeter ion simultaneously has both ionic states. Amino acids are example of sweeter ions. Figure 2.6. These compounds contain ammonium and a carboxylic group and can
Alpha amino acid are similar molecules. Why do amino acid in space favor the L conformation? Amino acid in space tend to favor L conformation due to a phenomenon called homochirality. This means that one enantiomers, in this case, the L conformation is overwhelmingly more abundant than its mirror image. While the exact reason for this preference are still being studied and no one really knows, but it is known that radiation can also exist in left and right-handed form. So, there is a theory called the Bonner hypothesis that process that the predominant forms of radiation in space from a rotation neutron star, for example, could lead to the selective formation of homochiral molecules such as L-amino acids and D sugars. This is still speculative, but recent findings from meteorites make this hypothesis much more plausible. Amino acids are zwitterions. In chemistry, zwitterions is a molecule with two or more functional groups, of which at least one has a positive and one has a negative electrical charge, and the net charge of the entire molecule is zero at a specific pH. Because they contain at least one positive and negative charge, zwitterions are also sometimes called inner salts. These the charge on the different functional groups balance each other out, and the molecule as a whole can be electrically neutral at a specific pH. The pH where this happens is known as the isoelectric point. Unlike simple amphoteric compound that may only form either cationic or ionic species, as deuterions simultaneously has both ionic state and amino acid are example of zwitterions in figure 2.6. This compound contain ammonium compound, ammonium and carboxylate group, and can be viewed as arising via kind of intramolecular acid-base reaction. The amine group deprotonates the carboxylic acid, and here's the figure 2.6. An amino acid contains both acidic carboxylic acid fragments and basic amine fragments. Centers the isomers in the rice is zwitterionic form. Because amino acids are zwitterions and several also contain the potential for ionization within their R groups. Their charge state in vivo and thus their reactivity can vary depending on the pH, temperature, and solvation status of the local microenvironment in which they are located. Chart of standard pK values for the amino acid is shown in Table 2.1 and can be used to predict the isonation charge status of any amino acid and the resulting peptides or the protein. And the pKa stands for the negative logarithm of the acid dissociation constant. However, it should be noted that the solvation status in microenvironment of amino acid can alter the relative pKa values of these functional groups and provide unique reactive properties within the active sites of enzymes, and that was shown in Table 2.1. As seen in Table 2.1, 7 of the amino acids contain R groups with ionizable with chain and are commonly found in the active site of enzymes. Recall that the pK is defined as the pH at which the ionized and unionized form of an ionizable functional group within a molecule exists in equal concentration. Thus, as a functional group shifts over or below its pK value, there will be a shift in the concentrations of the ionized and ionized form favoring one state over the other. Cytine and the disulfide bond formation. Cytine is also a unique amino acid. At this side, chain is capable of undergoing the reversible oxidation reduction or the redox reaction with other cytine residues, creating a covalent disulfide bond in the oxidized state figure in 2.9. Recall that when molecules become oxidized, that may are losing electrons, and that when molecules are reduced, that they are gaining electrons. During biological redox reaction, hydrogen's ions or the protons are often removed when the electrons from the molecule during oxidation and are returned during reduction. Thus, if a reaction is losing or gaining protons, this is a good indication that it also been losing or gaining electrons and that a redox reaction is occurring. Thus, proton gain or loss can be an easy way to identify this reaction time. Disulfide bonds are integral in the formation of the three-dimensional structure of proteins and can be therefore highly impact the function of the resulting protein. In cellular systems, this disulfide bond formation and or disruption is an enzyme mediated reaction and can be utilized as a mechanism to control the activity of protein. Disulfide bonds will be discussed in further detail section 2.xx within this chapter and in chapter XX.
As you can see on the upper right side, it is a downstream amino acid, and on the left side, it is a upstream amino acid. The downstream amino acids come from another amino acid, while upstream amino acids come from before it. So N is the protein beginning, H in the amide structure equal to peptide bond, R is the R group alternate in trans conformation, and the O is the next amino acid would be added here. Amide resonance structure. During amide resonance, the lone pair electrons from the nitrogen are involved in pi bond formation, with the carbonyl carbon forming the double bond. Thus, amide nitrogen are not basic. In addition, the carbon and nitrogen bond with the amide structure is fixed in space and cannot rotate due to pi bond character. So amide resonance means that the electrons from nitrogen and carbon together making a strong connection. This make amide nitrogen not act like bases. Also, the bond between carbon and nitrogen can twist because of strong connection. Instead, they are involved in pi bond formation with carbonyl carbon, so they join with the carbon atom in the bond to make a strong connection. Furthermore, the carbon to nitrogen bond within the amide structure is fixed in space and cannot rotate due to the pi bond character. So, the bond between carbon and nitrogen in the amide structure cannot twist or torn because of strong connection formed by pi bond. This creates fixed physical location of our group within the growing peptide in either the cis or trans conformations. Because the R group can be quite bulky, they usually alternate on either side of the growing protein chain in the trans conformation. This makes sure that R groups are certain places along the peptide chain, either facing the direction of opposite direction. Because some R groups are big, they often switch sides as the protein chain grows. This the cis conformation is only preferred with one specific amino acid, like chlorine. This is due to the cyclic structure of proline R group and the steric hindrance that is created when proline adopts the trans conformation. The cis shape is only linked by one amino acid, proline. This happened because of how proline shape is, and when it tries to be in the trans shape, it gets stuck because of how crowded it is. Thus, proline residues can be a large impact on the 3D structure and the resulting peptide. So, proline amino acids can greatly affect how the peptide ends up looking in 3 dimensions. Cis and trans conformation of amino acids are groups peptide bonds, cis or trans. So X and Y, this represents 2 consecutive amino acids in a polypeptide chain. X and Y are placeholder for the specific amino acids. In trans, this indicates that our group of amino acids, X and Y, are on opposite side of the peptide bond between them. In cis, this indicates that our groups of the amino acid X and Y are on the same side of the peptide bond between them. So, pro X pro refers to proline, an amino acid with a unique structure. When the preceding amino acid is in the trans conformation, the peptide bond between it and proline can adopt either a cis or trans conformation. So trans and cis indicates the possible configuration of the peptide bond when the preceding amino acid is in the trans conformation. It shows that despite the preceding amino acid being in the trans conformation, the peptide bowing involves proline can still adopt cis conformation more frequently due to steric hindrance caused by structure of proline. So overall, this explains how the presence of proline can influence the cis and trans conformation of peptide bonds in a polypeptide chain. Even the preceding amino acid is in that trans conformation.
Good day, everyone, and good day to our instructor. And for this continuation of the report of my co-reporter, so its topic about the diagram of the 2.12, so the cis and trans. As what we saw in the diagram of the 2.12, so we will tackle first what is cis and trans. So we'll recall it in our organic chemistry. The cis and trans are prefixes used in the chemistry to describe the arrangement of atoms, like what you saw in the diagram. It is a group of atoms around the double band. So in a cis configuration, similar groups are on the same side of the double band, while the trans configuration there are on the opposite side. So in the upper diagram of the figure 2.12, the conformation of amino acid R group, it displays a cis and trans conformation of two adjacent amino acid known as X and Y. So back to the diagram, we saw the X and Y. So in trans conformation, the R group of the amino acid X is rotated away, and on the other side of the molecule, when compared to the R group of amino acid Y, this conformation gives us at least amount of steric hindrance. Steric hindrance compared with the cis conformation, where the R groups are located to the same side close proximity to. So as the diagram, it is a peptide bonds. It is the covalent bonds that are linked to the amino acid together in a protein chain, and also they form between the amino acids, amino groups of one amino acid and carbonyl group of one another. So it will result to release of another water molecule. So in this process known as dehydration synthesis. So also these steric hindrance are also called as a steric strain, also called as steric hindrance. So it also occurs when the atoms in a molecule are forced into close proximity with each other, leading to repulsive interaction between their electron clouds. Also, this change result to the higher potential energy that can affect the stability and reactivity to the molecule. So, at what you see in the diagram of the 2.12, this is the steric strain, or also called the steric hindrance. So, in the other slide, so what is protein? So, proteins are a large complex of molecules that compose of chains of amino acid. They play a crucial role in the structure, formation, and regulation of the cells and tissues, and also in the living organism. So proteins are involved in the wide range of biological processes, including the enzyme catalysts, cell signaling, support of molecules, and structural support of immune response. So in the specific structure, function of the protein is very broad and also determined as a sequence of amino acid which fall into three-dimensional shape. So protein range in size of 50 amino acids and in length as largest known protein containing 33,423 and according to the slides. So when the largest, it is contained with 33,423 amino acid, while the fewer molecules, it is contained only 50 amino acids, also known as peptides. So as shown in the diagram, the figure 2.13, it is peptides and the proteins are macronutrients built from a long chain of amino acid joined together through amide linkage. So that is the amide linkage in the figure 2.13, and it is order to nature amino acid in the primary sequences of protein. So it will be surrounded by a very nonpolar hydrocarbon tail, like what you see in the figure 2.13. So, due to the large pool of amino acid, it can be incorporated with each position within the protein. So there are billion different possible protein combination that can be used to create novel protein structure. So there's a billion of possibility of combination that can create novel protein structure. So example of this is a tripeptide. So what is tripeptide? Before we read the PDF, the tripeptide is a molecule consisting of three amino acids. So it is linked together by peptide bonds. Like all peptides, tripeptides are formed through condensation reaction, where the amino group of one amino acid reacts to the carbonyl group of another. So it will release a water molecule, and also tripeptides have a various biological function depending on their amino acid sequence structure. For example, think like we will read the PDF. So think about the tripeptide made of the amino acid pole. At each position, there are 20 options that can be incorporated. Thus, number of resulting tripeptide possible will be 20 times 20 times 20, so equals of 8,000 different tripeptide options. So each of these options would vary in overall protein shape, as the nature of the amino acid chains help to determine the interaction of the protein with the residues in the protein itself and its surrounding environment. The character of the amino acid throughout the protein help the protein to form and form its three-dimensional structure. So it is this like I will read this PDF. So in this 3D shape that are required for the functional activity of the protein, it is example the protein shape equals a protein function. So, for the protein found inside the water environment, the cell hydrophobic amino acid and hydrophilic amino acid. So, what is hydrophobic and hydrophilic amino acid? So, before we will discuss. So, as the proteins are like molecular machines and their structure is crucial for their function, firstly, hydrophobic amino acid tends to avoid water because they don't interact well with it. So they tuck away inside the protein and shield them to the watery environment. So meanwhile, hydrophilic amino acids are attracted to water and prefer to interact with it. So they position on the protein surface when they are bound with water molecules. So when we say hydrophobic, it don't interact with the water, while hydrophilic amino acids are attracted with the water and prefer interact with its positions. So these arrangements help stabilize and protein structure and ensure its performance of its biological task effectively with the cell.
Once again, good morning everyone. So in continuation of the report of peptide bond formation and primary proline. As per stated in the module, proline, methionine, and cysteine play crucial roles in peptide bond formation and primary protein structure. So the proline's unique structure forms a rigid kink in the protein chain, impacting the protein's overall conformation. It has the only R group that forms a cyclic structure with the anime functional group in the main chain. This rigidity often affects protein folding and stability. For the methionine serves as the starting amino acid for almost all of the many thousands of proteins known in nature. Additionally, it provides a methyl group that can be involved in various biochemical reactions contributing to protein function. So for the cysteine is one of our body's 10 non-essential amino acids, meaning we can synthesize by ourselves. Cysteine play a very important role in our body as it stabilizing proteins, detoxification, and supporting different metabolic functions, and we can also find it in our skin, hair, and nails. Cysteine is unique. It's one of the only two amino acids that contains sulfur, and the only one has a thiol group, which essentially is a sulfur hydrogen bond. So now, back to disulfide bond. Disulfide bond are covalent bond that join two cysteine molecules. Based on the given pictures showing the disulfide bond, in the examples, it illustrates the two peptide chain that form the hormone insulin are depicted. As I said a while ago, disulfide bonds are formed between two cysteine residues, as you can see on the picture within a peptide or protein sequence, or between different peptide or protein chains. Disulfide bridges between the two chains that are required for the proper function of this hormone to regulate blood glucose levels. For the protein shape and function, the primary structure of each protein leads to the unique folding pattern that is characteristics for the specific protein. In the example in figure 2.15, thus the primary sequence is the linear order of the amino acids as they link together in the protein chain. To give you more knowledge about this, we can learn it in lesson 6 as they elucidate protein folding that give rise to secondary, tertiary, and sometimes quaternary protein structure.